Welcome to Bioratna. Dear students, today we are going to discuss the topic semi-conservative mode of DNA replication. Here there is one new word for you, DNA replication. What is replication? When an organism is reproducing at that time for its offspring, it needs another set of DNA. So what it has to do, it has to go through a process where it forms a replica, a copy of its own DNA and then gives it to the offspring. This is one thing, it is reproducing. Or it may be like a multicellular organism, a cell is dividing to form the daughter cells. Then the daughter cells, they also need their own set of DNA. Then the parental cell will form DNA, its own DNA replica and give it to the daughter cells. This is DNA replication. As the newly formed DNA is a replica, copy of the parental DNA, that's why it is called replication. It is also called duplication because by forming its replica, it is duplicating its DNA. So it is DNA replication. Now let us see what do we mean by semi-conservative mode. Look here. This is one strand of DNA. This is another strand of DNA. Here they have their nitrogen bases. One thing to be remembered, very important, is that, which you already know, that A always pairs with T with 2 hydrogen bonds and G always pairs with C with 3 hydrogen bonds. And these hydrogen bonds, they are very weak. They can be easily broken. Now, this is the parental DNA. This is the parental DNA. Now, this parental DNA has to form a replica for the offspring, which is such a huge work, as well as it is so much of responsibility so that the genetic information is passed properly. Here the 2 strands, they will separate from each other. So these 2 strands, they have separated. This one is C, this is T, this is A. This is G, this is A, and this is T. These hydrogen bonds are broken and they have separated. When they have separated, then now this plus this, I am putting a plus sign here so that you see that these are the 2 separated strands of the parental DNA. Now the whole information as it is there in the both strands, and this new strand also cannot go wrong because the new strand will be formed by the complementary rule, by which automatically you will see that the same molecule is formed here, and here also the one new strand which one is formed, it will also have both these molecules, this one and this one, these are exactly same to the parental strand, but here this strand is the conserved one. After this DNA replication is completed, then each newly formed DNA is having one parental strand, this is its parental strand, this is parental strand, and one newly formed strand. So this is called semi-conservative mode of DNA replication. Students, when Watson and Crick, they proposed the model of DNA in 1953, then looking at their structure, they said that the model which we have given, looking at it, we can see, we can hypothesize the mode by which this DNA might be replicating. At that time, proof was not there. Today, we are going to see the proof, how the proof was provided in 1958 by Meselson and Stahl. We are going to study that only. But in 1953 itself, Watson and Crick could hypothesize how, because they could see that these hydrogen bonds are easy to break, less energy expenditure, and then most importantly, it is just not simply an easy task, it is simply not a less energy, it is expenditure process. अगर काम करना है, एनर्जी बहुत खर्च करना है तो सेल करता है, but it can be avoided here. And most importantly, the whole information is intact in the two strands, and these two parental strands, they act as template strand. They act as template strand and they form the new strand by the complementary rule. What do we mean by template? Template is that strand which by its structure, it is physically able to direct the formation of a second structure. So this is the old structure which is directing the formation of another structure, as there is a complementary rule of CGAT like that in DNA. So this is semi-conservative mode of replication, and we will now see the how Meselson and Stahl, by a intelligent experiment, they proved that DNA replication is of semi-conservative type. Designing the experiment is the most important thing. You know about this Meselson Stahl experiment, you will appreciate the designing of the experiment. Now that you know what is semi-conservative mode of DNA replication, let us now see how Meselson and Stahl, they proved it in 1958. In 1958, Meselson and Stahl, they proved that DNA replication is semi-conservative. How? First, you need to know one thing. The DNA consists of hydrogen, oxygen, nitrogen and phosphorus, etc. The nitrogen which is present, normally which is present in our biomolecules, it is of N14 type. N14 type of nitrogen, atomic weight 14. There is another nitrogen which is a bit heavier one. This is N15. This N15 is a heavy, is an isotope of N14 which is heavier. Now, what Meselson and Stahl they did, they took E. coli, whatever E. coli they must have taken, everyone in their DNA it is having N14. Now they want the E. coli to have only N15. So what they will do, they for many generations they grew this E. coli on a medium which was having only N15, nothing else. NH4Cl was used, in which this N was N15. So after many generations, this N14 became so negligible that it was as if all the E. coli are having the heavy nitrogen only in their DNA. So here we are writing this is N15 and this is also N15. Both are having N15. Here now the experiment starts from this one. Okay, so let us see what happens. This is the parental DNA which is having N15 in both of its strands. सारे DNA में N15 ही incorporated है. Now this E. coli it reproduces after 20 minutes. 20 मिनट बाद. अच्छा, वन थिंग, यहाँ आपको ये लीनियर डीएनए दिख रहा है या इसके अराउंड सेल नहीं दिख रहा है, दैट डजंट मीन दैट दिस इज अ आइसोलेटेड डीएनए. नो, दिस इज बैक्टीरिया. ये जैसे एक सर्कुलर डीएनए ही है और it is inside the bacteria only. DNA cannot replicate without having the cytoplasm, those ribosome, etc., all the materials. So here we are showing DNA which is of significance for this experiment. So after 20 minutes, when this is reproducing, so if it is semi-conservative, so now these two DNA strands, parental DNA strands will separate. These two will separate and they will act as template. So this is N15 and this is N15. We have already seen what do we mean by template, but still I am repeating. Template is that structure, one structure which can guide the formation of another structure. इसका second strand कैसे बनाना है, इसको ये guide करेगा. But now what they have done, यहाँ पे अब जो medium available है, that is N14. Now it is only and only N14, no N15. तो अब जो नया स्ट्रैंड बनेगा उसके डीएनए में N15 नहीं रह सकता है, उसके डीएनए में रहेगा N14. So now the new strand which is formed, two colors are very much required. So the new strand which is formed, this new strand will have N14. This new strand here also it will have N. This is new one is N14. This is the after 20 minutes. The two N15 strands they have separated, and the new two strands which are formed are having N lighter one, that is N14 in their DNA, and then now after another 20 minutes what will happen, these four strands, now this will again replicate. The bacteria goes on reproducing in every 20 minutes. Here we have written 20, here let us write 40. So now these four will separate, each one will act as template. सिंपली सेपरेट करते हैं. चलो, दो 15 है और दो 14 है. ये हम लोगों ने ले लिया N14 और एक है N14, एक है N15 और एक है this one with blue color. And this one is N15, this one also N15. ये चार स्ट्रैंड सेपरेट हो गए हैं and the new strand which will be formed, this new strand will be N14 only. तो यहाँ इस N14 के साथ और एक N14 जुड़ गया, यहाँ इस N14 के साथ और एक N14 जुड़ गया. Here with this N15 it is one N14 and here this one new one, this is N14. So here you can see that these four strands which are formed, in them these two strands they are having all two strands, both the strands having N14, means this is the lighter one. This one it is having parental one जिससे start किया था, it is having both N15, means the heavy one. Here अब ये पेरेंटल बैक्टीरिया से ये दो बैक्टीरिया बने हैं ना, इनके दोनों के डीएनए सिर्फ हम लोग शो कर रहे हैं. सो इट इज हैविंग वन N15 वन N14, क्योंकि न्यू मीडियम में उसे N15 नहीं मिला है, N14 ही मिला है. नाउ एट ईच स्टेप इन्होंने ये पेरेंटल डीएनए को निकाला, फिर सीजियम क्लोराइड डेंसिटी ग्रेडियंट में सेंट्रीफ्यूगेशन किया. then they settle according to their density, वो heavy है या ऐसा medium है या ऐसा light है. Accordingly they will settle at different planes ना. So this one is having only heavy one. इसके दोनों के दोनों जो strand है, दोनों strand जो एक दूसरे से लिपटे हुए हैं, both are in 15. तो अगर suppose ये वाला zone है heavy वाले का settle होने का, तो you will see only this band and no other band. इसमें सिर्फ आपको यहाँ पे बना हुआ एक बैंड दिखेगा which is for heavy band. At this stage when they separated and did this cesium chloride density centrifugation, then अब देखिए आप ये दोनों स्ट्रैंड में से हर किसी के पास एक हेवी है एक लाइट है, एक N14 है एक N15 है, मीन्स बोथ आर हाइब्रिड. तो अगर ये वाला पोजीशन हम लेते हैं हेवी का, ये लेते हैं मीडियम वाला मतलब हाइब्रिड वाला, और दोनों N14 का लाइट वाला यहाँ अगर बनेगा, तो इसमें आपको इस जोन में कुछ नहीं दिखाई देगा, हेवी में, लाइट में भी कुछ नहीं दिखाई देगा, बिकॉज बोथ आर hybrids, so they will form a band at the medium density level here. Now come to the next one. After 40 minutes, these two bacteria, they have multiplied, their two DNA strands have separated, acted as template, and then for the new DNA they have taken the N14 one. So अब आप देखिए इसको, इसके पास N14 वाला दो है, मतलब दोनों लाइट है, both N14 they are the light one. तो ये दो लाइट है. अब हेवी वाला अगर बैंड यहाँ बनना है, मीडियम वाला यहाँ बनना है, लाइट वाला यहाँ बनना है, लाइट वाला बनेगा क्या? हाँ बनेगा, यहाँ दो लाइट है. सो वन बैंड यू कैन सी हियर, एंड हेवी है क्या इसमें? कहीं नहीं है, यहाँ हेवी में कुछ नहीं बनना है. और मीडियम वाला है क्या? दिस इज द हाइब्रिड वन. सो हियर ऑल्सो यू कैन सी द, this is the light one, this is the hybrid one which is having medium density, and this hybrid नहीं लिखते हैं यहाँ, दोनों H हो जाएगा, this is H and this is the medium one. So this medium one. So this type of band can be obtained only and only when the DNA replication is of semi-conservative method. क्यों? अगर इसने अपने दोनों स्ट्रैंड अपने पास रख लिए होते और उसने दोनों दूसरे स्ट्रैंड नए बनाए होते, तो आपको यहाँ पे इसका हेवी वाला एक में दोनों हेवी रहते तो आपको हेवी में भी मिल जाता, और दूसरे में दोनों नए रहते तो मीडियम वाले में कुछ नहीं मिलता. दोनों के दो, फिर एक आपको हेवी में मिलता पेरेंटल वाला, और नया बना हुआ N14 वो आपको यहाँ लाइट में मिलता. But that is not the case. So this conclusively proves that DNA replication is of semi-conservative nature. इसके बाद इसी समय 1958 में ही there was another scientist, Taylor, and his colleagues. They also proved that DNA replication is a semi-conservative one, but they did it with the help of thymidine, radioactive thymidine they used, and the experiment was on the plant Vicia faba beans. This is how it, you can see how interesting biology is, and how just take interest, and all topics will go on unfolding. That's it. Join, subscribe. See you in the next video.